Okay. So can you see my slides now? Let me just project this one. So we will not really go slide per slide, okay? Hello. So we will not we will not be reading all the slides. Now we'll just run through because we have around 90 slides to finish. No, so if you think that I'm going too fast, just inform me so I can also make the necessary adjustments. No, so we will be discussing again module 7, 8, and 9. Okay. Before we go to that one, let me check if okay, it's the lecture today is recorded so that we can also share this to those who are not able to attend. Okay, so last week I told you that I will be giving you a quiz, no? But I I have to modify it in such a way that those who will not be able to, those who are not here, no, will not be disadvantaged, because I want you to understand that we had a memo, no, the faculty members were given a memorandum and a reminder that we should not give graded activities during synchronous classes. So ganun yun. So those who are not capable or can't connect online and join the synchronous class will not be disadvantaged. Have you noticed, masyadong mabait ang system ngayon sa mga students, no? Swerte-swerte nyo naman. Hindi, mabait sila sa inyo, mabait din naman sila sa amin. So this module will discuss about fish spoilage, the types of microbial fish spoilage, the factors influencing the rate of spoilage, and the control of spoilage. May I ask, meron ba sa inyo dito na enrolled sa Fisheries 160? Meron? Is there any one of you here is also enrolled in Fish 160? Wala. Okay. Fisheries 160 is a course code for fish handling and preservation of products. And in that course, we're talking a lot about fish spoilage, no? Post-mortem changes, and everything related to spoilage and handling. Ayan. So, food and food spoilage, there are around 25% of all food produced in the world is lost due to post-harvest, post-harvest handling, and that is acted on by microbes or microorganisms. Ayan, be it the microorganism itself or the enzymes produced by the microorganism. Okay, so food are classified into perishable, semi-perishable, and non-perishable. So what do you think are the characteristics or attributes of the food that makes them, or that made them classified under these categories? So for meats, fish, poultry, eggs, and milk, most of these they have high water activity. So the water activity, the pH of the food, and other factors or other attributes are the ones that make them be classified under perishable, semi-perishable, or non-perishable. So what do you notice about potatoes, apples, and nuts? They are dried. So they have low water content or moisture content. That's why they are semi-perishable. And for this flour, sugar, rice, dry beans, they are very, very dry. They have very low water activity. It's not just about moisture content, but water activity at the same time. Water activity is more important. Is a more important attribute to look at in foods when you talk about spoilage, especially if it concerns microbial spoilage, because the water activity is the water used by microorganisms for their growth. Okay. Sige, so we have here the definition of spoilage. So I guess you have already read this one. This can be defined as a change in fish or fish products that renders them less acceptable or unacceptable or unsafe for human consumption. And so during the spoilage process, there is a breakdown of various components, no, in the formation of new compounds. And take note that these new compounds are used by the microorganisms, no, they use the new compounds as a product of spoilage as their food for further growth. So mas magpaparami sila they utilize the breakdown products in fish. Later you will know that there are a certain group of microorganisms classified as SSOs, in which they produce certain compounds that makes that results to undesirable sensory characteristics of the fish or food. Okay. These new compounds are responsible for the changes. Yung mga new compounds produced during spoilage are responsible for the changes in the odor, flavor, and texture of the fish meat. Okay. So how would we know, that the fish is already undergoing spoilage? What are the signs? So we have here the signs, and what are the agents? So the agents could be the proteolytic enzymes, and the proteolytic enzymes are the one responsible in the autolysis, self-degradation of proteins in fish. And then the chemical or enzymatic, it could be caused by the indigenous enzymes in fish. Ibig sabihin yung enzymes present in the fish tissues, or could be caused by the enzymes produced by the microorganisms. And we also have microbiological activity. So these are the signs of fish spoilage. I think we don't have a table here showing or comparing the characteristics of a fresh and spoiled fish, but here are the signs of spoiling fish. Slime starts to form. There will be discoloration, especially in the case of salmon, for example. Diba, if it's very, very fresh, it's bright red. But if it's already starting to get spoiled, the color becomes pale red until maging very, very pale na almost white. Okay. And also there will be changes in texture, development of off odors and flavor, and also gas production. All right. So this figure shows it's quite complicated at first glance, right? But it simply tells us that at pre-rigor, do you know what rigor mortis is? Can anybody share in the class what rigor mortis is?
Sa the explanation naman, rigor mortis, yung pagtiskog sa isda, ma'am.
Okay, very good. So, rigor mortis refers to the stiffening of the muscle of not just fish, no? Bisan ang tao kung mapatay, it undergoes rigor mortis, no? And upon, diba, while the fish is still living, its tissue, muscle tissue is still sterile, meaning sterile siya. And then upon death, medyo soft pa ang fish. Okay, soft ang fish, it undergoes a pre-rigor stage. When you say pre-rigor, it is in a relaxed state, soft siya. And then after some time, the fish muscle will start to contract and stiffen, no? So that state is already called the rigor mortis stage. Okay, so pag nag-stiffen, naninigas, that's the rigor mortis stage. And after some time, that would depend on the species. The muscle tissue or the muscle of the fish will go back to a relaxed state. Okay, so what triggers the contraction or the stiffening of the muscle? So the absence of, or di ba, upon death, the supply of oxygen in the fish tissue will drop. Okay, so the production of ATP will also stop. So that's obvious, right? And at that stage, no, there will be cessation of the production of oxygenated blood. There will be a lot of ATP that will cause the continuation of the normal processes in the fish muscle. So during that stage, or the tissue will now shift to anaerobic glycolysis, and that glycolysis would result in the production or accumulation of lactic acid, which will make the fish muscle acidic. So what are the factors that contribute again to the stiffening of the muscle? So look at this. Can you see my cursor? Can you see? Can you see it?
Yes, ma'am.
Okay, so in the stiffening of the muscle, there are factors that affects the stiffening. All right. So we have the actin and the myosin filament. So I don't know. I think wala na include there is a slide nga ni ang sliding filament theory, no? But at a relaxed state, no? Pag meron pang ATP, the calcium ions will be pumped into the sarcomere, and there will be sliding of the actin, no, actin and filament, actin and myosin. So this is the relaxed state. Look at this, the actin are away from each other, and this is your myosin, the red thing, no? But at a contracted muscle, the actin are drawn closer to each other, like this one. Mas malapit na yung mga actin filament, and that triggers the contraction of the muscle. So calcium here has a participation, no, in the formation of the actin-actomyosin complex during the contraction of the muscle. Now, since paubos na yung, imagine niyo na pag-relax siya, madami siyang ATP, and during the rigor mortis state, paubos na yung ATP, right? So, wala nang there's no more energy to fuel the processes in the cell for the calcium ions to be pumped out. So, ang mangyayari niyan, di ba, sabi ko kanina, there will be a relaxed pre-rigor state. There is a rigor mortis state wherein nagsistifan yung muscle ng fish, and after that rigor mortis state, babalik siya dun sa relaxation state. So at the time na matatapos na yung rigor mortis state, magre-relax ulit yung muscle, it's an indication that there is already nag-undergo na ng spoilage yung fish natin. Okay, so you just read the guide here, no? So you will learn more about the stiffening or the actin myosin complex formation. Okay, and there is a deeper explanation, no, on the contraction of the muscle or the stiffening of the muscle of the fish, but that is already beyond the scope of this course, no? Pag nag-MS kayo, you will learn more about rigor mortis, okay? So at least you know that pag namatay yung isa, maka-cut off yung supply ng oxygen, the ATP production will also be lessened to the point na totally drop, no? And then there will be it will now the mesh tissue or the muscle will now go into a rigor mortis state, and after some time it will relax again, and that's the time na mag-a-attack na yung spoilage enzymes, microorganisms, and other processes will take place until mag-spoil yung fish natin. Okay? So again, autolysis takes place, and there is this chemical oxidation. The chemical oxidation really takes place in fish, no? Especially that we know in our past modules that or we know that fish contains a lot of polyunsaturated fatty acids. Do you agree that fish has high amounts of polyunsaturated fatty acids? Okay. Though the high amounts of PUFA, we call it PUFA in fish, is very advantageous because PUFA are healthy lipids. Okay, but they are very unstable, and the downside of it is very prone to oxidative spoilage, or madali silang react with other substances, very reactive, and that would lead to the formation of odors and flavors in fish, especially in fatty fish species. Okay, and another one is the microbial spoilage. Okay, so this slide shows the 4 phases of fish spoilage, no? So if you are familiar with sensory evaluation, if we are doing face-to-face, no, in the lab, we will give you samples of food products or fishery products, and then we will give you a sensory scorecard. So in that scorecard, as you taste or test the product, you will rate the product according to its sensory score. No, for example, you rate the odor. If it is fishy or if it's ammoniacal or something, you put a score. And then if you are asked to evaluate the flavor, you also have to indicate whether it is sweet fishy flavor, it has a sweet fishy flavor or something else, no? You also have to rate, and also you have to check the texture and other attributes of the product. So that's how quality scores are generated. And this one shows the phases of spoilage, no? Okay, so during autolytic spoilage caused by enzymes, fish just caught is very fresh and has sweet, seaweedy, and delicate taste. And there is very little deterioration during phase 1. And in phase 2, there is already a significant loss of natural flavor, but the fish or the product is still acceptable. And in phase 3, no, there will be bacteriological changes in fish caused mainly by bacteria. And at this phase, the fish begins to show signs of spoilage, while in fourth phase, no, fish is spoiled and putrid and becomes inedible and unsafe for consumption, unsafe for consumption na siya. Okay? So you can actually make an experiment, no? And then determine, kasi dito yung days of storage. So you can actually determine at what particular day, for example, if you store your product in ice, how many days stored in ice yung fish mo before siya maging spoiled. And ilan yung sensory score niya para maging spoiled siya. Para ma-detect mo na it already has an unacceptable flavor and odor. So ito yung rejection limit. No, rejection limit for this particular species. So I guess this figure was taken from FAO, no? And I'm not sure if the fish sample for this graph is a cod, no? Okay, anyway, so that's how you create your own graph. Okay, so for fish spoilage, teka, I'll check my time. So dapat meron tayong maximum of 45 minutes lang per Ay, hindi siya 45 minutes. Sana 30 minutes per module. Okay, so you just read this one about autolytic enzyme spoilage. The most important process during autolytic enzyme spoilage is the degradation of nucleotides. So in this figure, it shows the degradation process of um, adenosine triphosphate or ATP, in which ATP is broken down to IMP, ATP to ADP, adenosine triphosphate, adenosine diphosphate, adenosine monophosphate, and then inosine, hypoxanthine, and so on. So, what are the important uh, degradation products in this figure? So you have to note that IMP here, IMP is responsible for the, please correct this one, HX is not responsible for the sweet and mild, sige, check ko na, i-edit ko na ngayon. Ayan. Ayan. Mali ito, no? Sino ba yung gumawa ng slide na to? HX and inosine are responsible for the bitter taste of spoiled fish. Ayan. Balik tayo doon sa ating, Ayan. So take note that IMP, inosine monophosphate, is responsible for the sweet, mild taste in fish. However, accumulation of this IMP is very quick, no? And these products are the ones responsible for the bitter taste of spoiled fish. So the amount of your IMP, inosine, hypoxanthine, and all of this will actually depend on the amount of ATP as a starting material in the spoilage process or in the degradation process. Do you agree? So you can actually measure the degree of spoilage by measuring the degradation products, inosine and hypoxanthine, no, over all of its components. So that portion naman is discussed in, this concept is actually discussed in more detail sa Fisheries 155. So at least for our purpose, you know that ATP is degraded into IMP and inosine, hypoxanthine, and to uric acid as a final product. And you know that IMP is the one responsible for the sweet and mild taste in fish, and the inosine and hypoxanthine are the ones responsible for the bitter taste in spoiled fish. Okay? Ayan. Autolytic changes, enzyme spoilage, precede microbial or microbiological spoilage and have been seen to contribute very little to spoilage of chilled fish and fish products. Bakit very little? Kasi at chilled condition, suppressed ang action or ang activity ng enzymes. Okay? And autolytic enzymes reduce textural quality during early stages of deterioration but do not produce characteristic spoilage of odors and flavors. No, hindi yung enzymes ang nagpo-produce nito ng mga of odors and flavors. And the digestive enzymes cause extensive autolysis, which results in the meat softening, rupture of the belly wall, and drain out of the blood water, which contains both the protein and the oil. So yung mga lipases are responsible for the degradation of the oil, and the proteases ang irresponsible naman for the proteins. Okay? So for oxidative spoilage, no? Oxidation typically involves the reaction of oxygen. So that's why for you to suppress or to reduce oxidative or oxidation of your fishery products, you have to apply certain methods in your packaging. So for example, medyo fatty yung raw mat mo, you might want to reduce the amount of oxygen in your packaging by using a modified atmospheric packaging system or yung MAP na tinatawag, or you might also want to use vacuum packaging. So ganon, you will reduce the amount of oxygen in the pack, and at the same time, lower the oxidation potential of the product. Ayan. I am trying to discuss very briefly each slide. Medyo na ano ako, na pressure ako sa time. Okay? So for this one naman, no, this shows the lipid oxidation process from the unsaturated fatty acids or the PUFA. In the PUFA, in the presence of oxygen, will react, no, and produce these byproducts which are responsible in the production of odors and flavors in fish. All right. So if your product is subjected to high temperatures, no, that would trigger further, no, the oxidation process. Okay. So you just watch this video on your own. Okay. Can you still follow, guys? So for the microbial spoilage, no, we know that fish has microorganisms found on all the outer surfaces of its body, no. So and it also has microorganisms in its alimentary tract. And the normal flora of fish is around, or the normal range of microorganisms in the skin of fish is around 10⁻² to 10⁻⁷ colony forming units, no, per centimeter. And in its gills and intestines, medyo mataas, up to 10⁻⁹ CFU per gram. Okay? And when the fish dies, its entire body resistance mechanisms break down, giving way to microorganisms to invade or diffuse into the flesh. So yung sinabi ko kanina that fish, while it's still living, no, its muscle is sterile because it still has resistance mechanisms against microorganisms. Okay, so as mentioned, no, medyo mataas yung number of microorganisms in the surface of the fish when it's still living, but only a portion of it causes spoilage. So yun yung tinatawag natin na specific spoilage organisms. So fish microflora includes bacterial species such as Pseudomonas, and all of this, no, and they are involved in the succession of microbes. So for cold tolerant fish, no, or yung fishes that were collected from a temperate water, the genera that's present in its that's present in the fish are this one, while for fishes that have been collected from tropical waters have mesophilic spoilage bacteria, and that make the tropical fish spoil much faster than temperate water fish. Ayan. So pag hindi mo siya in-ice, ito ganito yon. If you subject or if you store your, for example, you're doing you are comparing the efficacy of icing in retarding microbial spoilage in your fish. Ano yung mas mauunang mag-spoil? Yung temperate fish ba or yung tropical fish? If you do icing or chilling, ano yung mauunang mag-spoil? Temperate fish or yung tropical fish? Any
Temperate,
yes.
ma'am. Temperate fish, ma'am.
Okay, why is it that temperate fish will spoil faster in ice?
May bacteria, ma'am, na mga cyclophilic, ma'am, na present sa temperate fish.
Okay, so very good. Sino yon? May I know sino yung sumagot?
Julian, ma'am.
Hi, Julian. Okay,
Hi,
so
ma'am.
that's tumpak. Very good, Julian. Okay, so there are cyclophilic microorganisms present in temperate fish. So remember, kumbaga yung ice mo walang effect doon sa mga cyclophiles. Okay, whereas yung ice mo has tremendous effect doon sa mesophilic microorganisms kasi hindi nila gusto yung cold. Gusto nila around 20 to 37 degrees Celsius na temperature. Okay? So, yun yun. Alright, so this one shows the parang simple representation on how specific spoilage microorganism cause spoilage in fish. Parang paulit-ulit na nakakabingi yung spoilage na word. But ito kasi yung coverage ng ating module 7. So we have to deal with it. Ayan. Microorganisms are capable of producing enzymes, and that enzyme could be amylases, lipases, and proteases. So our diba, these types of enzymes attack on specific substrates. Itong amylases sa carbohydrates, lipases sa lipids, and yung proteases sa proteins. Okay? And your dead fish will now serve as a substrate for spoilage organisms. Pag sinabi natin substrate, yun yung nagsiserve as food nila, no? So this will now result to the acidification of the muscle tissue of fish, and there will now be changes in its flesh, texture, and appearance. So pag acidic na siya, mag-change na yung color, mag-change yung texture, no? And also, it could cause changes in fish flesh. Later, you will see the definition of specific spoilage organism, that they are the ones responsible in producing metabolites. And these metabolites produced by these SSOs are the ones that triggers the sensorial changes in fish. Okay? This one naman, it's another complex figure, no? I'm sorry. But thanks to these notes that tells us about everything about this figure, okay? So we have here the spoilage microorganisms acting on the tissue of the fish, no? Specifically on the protein. So this one shows the protein degradation pathway, okay? And your protein will be degraded into shorter chains of shorter chains and into finally into amino acids. Okay, in this spoilage microorganisms will again produce the metabolites and will lead to the production of these keto acids. So mahaba pa yung lalakbayin ng protein. It will still undergo a lot of processes like the deamination, transamination, and so on, no? And this will lead to the formation of keto acids, and the other compounds that's related to the production of odor and flavor. So ito naman yung nag-show ng mga compounds related to the production of odors, and this one results to the shows the formation of the biogenic amines. So ito sila the formation of the putrescine, histamine, and cadaverine are dependent on the amount of ornithine, lysine, and histidine, no, in your fish. So kung mataas, for example, yung histidine, it is most likely that your fish will produce or form high amounts of histamine. So sinabi dito, biogenic amines cause off odors and threat to food safety. Bakit food safety? Kasi high amounts of histamine and this could cause allergic reactions to sensitive individuals. Okay? So, this also shows parang lang siya, repetition ng previous slides. Yun. At sinabi ko kanina yung mga by the way, let me go back here. So, yun, yung sinabi ko na alpha keto acids produces of odors and flavors in fish. And one of the end products are your ammonia. And one of the best means to measure the volatile base products, no, during fish spoilage is the TVBN assay, or the total volatile base nitrogen assay. You'll be able to measure all the amines produced, no, during fish spoilage. So ito yun. You determine the TVBN content, or the total volatile base nitrogen content. Okay? So ito yun. It also shows the specific spoilage bacteria or organism. These are the SSOs, and these are the spoilage compounds that they produce. So marami sa kanila ang trimethylamine, no? So ito yung mga dinedetermine natin using the TVBN assay. Okay? Ayan. This shows the how the spoilage bacteria act, no, on the products. High ba, mabilis, moderate, or low? So these are just the this shows the definitions, no, of the different types of microbial spoilage. So we have the putrefaction, and then you have the fermentation. We have the rancidity. Okay, and here are the factors influencing the rate of spoilage. So rough handling will of course cause damage in your fish, no? And high initial load of microorganisms in your fish would most likely cause fast fast degradation or quick spoilage of your product of fish. Okay, and this one shows the different methods of capturing fish and the consequences. So you'll know na kapag trawling yung ginamit mo, no? Mabilis yung spoilage because ang daming trawling causes physical damage dun sa fish. Okay, I will no longer elaborate this one, no? Kasi it's very easy naman to understand. Okay. Ayan, mode of storage. So, depende on how you store your product. If in bulk storage, the weight of the pile may crush the fish at the bottom, leading to a loss of weight, as well as other physical damages. All right. Crushing of the fish by ice or other fish can seriously affect the quality of fish by releasing enzymes. Take note na kapag may mga bruises or may mga physical damages sa fish, hindi lang basta na damage yung fish, kundi it also triggers the release of the proteolytic and lipolytic enzymes, no, that would lead to the degradation of the fats and proteins. Fish. Okay. So what are the examples of spoilage control? So prevention of the initial contamination. So if you know, no, if you can reduce the number of microorganisms at the start pa lang, better do it, no, because more organisms present in your raw material would mean shorter shelf life for your product. Okay, and low temperature preservation like chilling and freezing can reduce means the reduction of the temperature of your product, no, and this could may not totally stop enzymatic and microbial action, but at least it can reduce to a significant level, no. But take note that even during low temperature storage, no, kahit -40, -60, -80, there would still be quality damage or damages that will take place sa inyong product. Okay? Walang perfect. I'm so sorry to say that one. Okay, for processing, ayan, processing methods that allows the water content or the water activity of fish to be reduced, no, would also limit the growth of bacteria. Ayan, so we're done with this one. Okay, maybe we just proceed, no, with the next module? Do you have any question? Ay, teka, hindi ko siya pala na-open. Wait. Do you have any question, guys?
Ma'am, sa activity, bala ma'am sa last part, ma'am, i-submit pa to siya, ma'am, no? Yung group activity.
From 7, 8, and 9, no more activity, no more output.
Okay, ma'am. Thank you.
Oo, gusto niyo pa, Tani? Nabitin ka mo?
Hindi, ma'am.
Thank you, ma'am.
Yes, of course. Kasi parang na-shock din ako. Class, the reason why I reduced the number of outputs because parang na-shock ako na I thought we still have another week or two para sa klase natin. But nung middle of May or towards the end of the month, parang binigyan lang kami ng memo na until June 11 na lang yung class. So, ibig sabihin, last meeting na natin to. And ayoko rin namang maging unfair na tambakan kayo ng output na yung kumbaga unrealistic na, hindi na rin kaya ng time na considering we only have 2 hours per week and then you'll be doing your output for 'di ba? More than 2 hours. Magsa-study pa kayo ng PowerPoint tapos mag-output pa kayo. If you are taking 5 courses at ganun lahat. So, 'di ba, paano na lang ang mental health natin? And I am also thinking of myself. Siyempre, ganun din. Kasi, kung puro tayo output, hindi na ako makakapag-lecture. Yung ganun ba? Minsan kasi may mga inputs from your teacher na hindi nyo siya makikita sa slides. Okay? And, sayang naman yun. Once lang kayo magte-take ng Fisheries 154. Once lang dapat kayo mag-take ng Fisheries 154. Okay? Teka lang, class. Bakit parang na-out ako? Teka lang. Bakit meron akong Zoom dito na isa? Teka lang ah. Ay, hindi ko siya pwedeng i-X. Teka, class ah. Sorry. Bakit ako meron akong isang Zoom na naka-open. Okay. So, can you see my slide now? Itong microbial control and detection.
Yes, ma'am.
Ayan. This is one of the most interesting lecture na medyo favorite ko siya. Teka. This is module 8. Sandali ah. Bakit hindi ko siya ma-open? Anyway. All right. This slide is very is very interesting. No? If you're going to think if there will be a war between man and microbes, no? Feeling yung ano yung mananalo? If you will just physically fight against these microorganisms. 'Di ba? We will not be able to defeat them if we will not use our intelligence. So thank you to our ancestors. Yung mga minention ninyo na mga prominent figures in microbiology. So during our first module, no, na 'di ba? They contributed a lot to what we to the microbiology idea that we are enjoying now. No, kasi na-solve na yung problem dun sa mga microbes and may mga discoveries sila that up to this time no remains useful to all of us. Okay? So kaya na nating labanan lahat ng virus and lahat ng microorganisms including the virus and hopefully yung mga vaccine natin ngayon can really fight against COVID-19. Sino sa inyo nakapagpa-vaccine na? Meron na bang nakapagpabaksin?
Wala pa po.
Hindi rin kayo nakapabaksin. Pareho tayo.
Ginauna pa, ma'am, ang mga may comorbidities.
Oo, comorbidities. Oo. So at this time, di ba dati, we are very thankful that wala kita yasang sakit nga iban. But sa subong, no, para mambal ka po nga kadugay ako yung dugay ako yung i-vaccinate ya kay wala ko ya ano ya, wala ko ya comorbidity. So naging disadvantage pa subong nga wala ka comorbidity. And it's ironic, right? Anyway, sige. Wala kita sang mahimo. Okay? So what is microbial control? Actually, this slide, ang module 8 is very easy to understand, no? And I wanna give you an activity after this one, and hopefully we still have time for this. We are supposed to end at 3 o'clock, so dapat mga 20 minutes lang ini. Okay? So control of microbial growth means to inhibit or prevent the growth of microorganisms. So there are two ways, no, to do this: by killing directly the microorganisms or by inhibiting the growth of these microorganisms. So how would you inhibit the growth of microorganisms? So you can do that by interfering with the bacterial proteins production, DNA replication, or other aspects of bacterial cellular metabolism. So with that, if you inhibit DNA replication, no, hindi na sila dadami. Yung microorganism, it will no longer grow. Ano yung pinagkaiba nung killing the microorganism from stopping or slowing down the growth? Yung pinagkaiba nun, ako na lang yung sasagot para mabilis kasi 20 minutes na lang. Okay, if you kill directly, namamatay. Okay, lahat patay na. But if you inhibit or slows down the growth, ibig sabihin, pwedeng yung existing microorganisms will remain alive, but it will no longer increase in number. But eventually, pwedeng mamatay na rin yung mga pre-existing ng mga microorganisms. So that's the difference, no, between killing and inhibiting the growth. Kasi when you inhibit the growth, you don't allow the microorganisms to increase in number. Okay? So let's learn some important terminologies. Okay? Pag yung word has cidal, ibig sabihin it kills the cells. No, if it has static, it inhibits the growth of cells without killing them. So, na-appreciate nyo na yung value ng previous slide. So now you can define bactericidal and the bacteriostatic. Okay? So who are our enemies? Our enemies are the pathogens which affects our safety, no, and the spoilage microorganisms which affects the quality of our products. Okay, so what are the methods of microbial control? We have the physical, the chemical, biological, and the mechanical removal. So pag ni-remove mo, yung parang kinukuha mo lang talaga siya. So that's the example of that for that is the filtration process. Whereas yung biological methods, you use antibiotics. For the chemical methods, you use the antiseptics, disinfectants, preservatives. And for the physical, you apply heat or radiation. So you just read this one, no? These are the physical agents that we use, no? The moist heat, the under that one, pasteurization and autoclaving or sterilization. Take note that autoclaving is the most important, the most effective moist heat method. Yan. I have discussed this one in my I think in my lab class, maswerte yung mga estudyante ko sa lab, meron tayong ganito. Alright? So, moist heat is more efficient in killing microorganisms versus the air of the same temperature. Mas madali siyang makapatay yung moist heat. Okay? Under physical agents, we also have dry heat, freezing, and desiccation. Okay? Hindi ko na i-elaborate. For the radiation naman, no, with the use of ionizing radiation, okay, and ultraviolet radiation, and also microwave. Bakit sinasabi dito? Usually, microwave do not directly kill the organisms. It indirectly from heat generated in microwave material. So, ang ginagawa kasi sa microwave, no, pag andun yung food mo, nilagay mo sa microwave, microwaves will heat up the food material, and that the heat in the food material will now will cause protein denaturation and DNA denaturation sa bacteria, or it will denature the DNA of your bacteria na present sa food. So kaya siya sinasabing indirect yung killing ng microwave. Okay, we have the chemical agents, we have the disinfectants, antiseptics, and the preservatives. So basahin nyo na lang yan dito, okay? So, sige na nga lang. Disinfectants for inanimate objects, antiseptics for living tissues, and preservatives are added to the products. Okay, to prevent decomposition by microbial growth. Okay, so we have here the disinfectants, no? The bleach kills microbes by inhibiting the enzyme activity and oxidizing cellular content so that they no longer perform normal metabolic functions. Take note, hindi niya pinapatay, no? It reacts with the organic material in the cell it is used up. Pag nag-react yung chlorine, no, sa organic material in the cell, para siya nag-evaporate or it's being used up. So its concentration is nagdi-dilute siya, bumabawas. So its efficacy also decreases kapag nag-react na yung material sa organic components of your bacteria. So that's why for the chlorine to be effective, it should be used in high concentration. Ayan, sinabi dito. Okay? Kasi na-used up siya. So we have also the non-bleach, no? Example of that is your Lysol. And the mode of action for that one is they exert germicidal effect by denaturing the proteins and destroying the selective permeability of the cell membrane. So, diba, pag nasira yung cell membrane, it will now cause leakage of the cellular components. So, lalabas yung lahat ng components, and then eventually it will cause the death of your microorganism. We also have alcohol. and they denature proteins and dissolve the lipids with optimal concentration at 70%. Okay, I have a very interesting question. At ang mga nasa klase ko sa lab can answer this one. What's the difference of ethanol and isopropyl alcohol? Sige, walang sasagot. I will ask that one sa exam. At least alam nyo na, no? May isang question ako itatanong sa exam na ganyan. Okay, so which one is more effective? Kasi sinabi na dito na optimum concentration is 70% of the alcohol. So what is the difference in the action of 70% alcohol versus the 90% or the absolute or yung 100% na concentration ng alcohol? Anybody who can share? Sinabi kasi mas effective yung mas optimum na concentration 70%. Anybody who can share?
Mas dasig mag-evaporate ang 100%.
Okay, very good. That's one. Another one. Sino to siya gani,
Feeling ma'am, Luda.
When you, class, kay hindi ko makitaan bala ang names nyo kung gahambal ka mo. When you recite, please mention first your name, okay? Thank you. Okay, what else? Thank you, Feline. Aside from mas dasig mag-evaporate, so if it evaporates very quickly, meaning hindi na siya maka-act dun sa microorganism kasi nag-evaporate na siya, nawala na siya. Okay, that's good. What else? Ang concentrated na alcohol upon application dun sa microorganism, pag nag-touch, kumbaga, yung alcohol dun sa cellular, sa cell wall ng bacteria, for example, it will immediately results to the aggregation of the components. So, ang tendency, magko-close yung cell wall, hindi na makapenetrate further kasi sobrang concentrated. It can no longer penetrate the interior portion of the cell. So, hindi niya ma-target, hindi niya abot yung important cellular components, the DNA and the other components of the cell. And the bacteria itself can produce a protective coating para hindi na siya mapasok ng concentrated alcohol kasi nga mabilis yung action. But sa sobrang bilis, nagkukos siya ngayon ng further protection kasi nga hindi makapasok yung concentrated alcohol. Whereas the 70% alcohol slowly penetrates the cell wall and the interior portion of the cell, and it can, kasi marireach niya talaga yung mga cellular components including the DNA, so it can denature the proteins, dissolve the lipids, kasi diba we know that alcohols dissolves the lipids, right? And it can eventually kill the bacteria or the microorganism. So that's why lower concentration of alcohols are commonly used as a disinfectant or antiseptic agent. But very low concentration is no longer is also nagiging less effective na rin siya, especially if the concentration of alcohol drops to as low as 40% and below. Okay? So that's all for alcohol. We also have these super surface active ingredients or surfactants. We also have antiseptic compounds. And we have these antimicrobial agents. Ayan. So antibiotics, also known as the antibacterials, are medications that destroy or slow down the growth of bacteria, and they include a range of powerful drugs and are used to treat diseases caused by bacteria. And these antibiotics cannot however, these antibiotics cannot treat viral infections such as cold, flu, and other coughs, mga ubo-ubo. Ayan, so as we've mentioned earlier, the modes of action of these antimicrobial agents can be of two categories: alteration of cell walls or cytoplasmic membrane, babaguhin niya yung cell wall, or interference with the protein and nucleic acid cycle. So pwedeng babaguhin niya para ma-penetrate ng antimicrobial agent yung contents, or mag-i-interfere siya dun sa protein and nucleic acid structure. Ayan, we will be having an activity in a while, no? We will use this figure. Sige, at tabayanan natin. At tabayanan nyo, tapusin lang natin itong lecture na to. So, for the alteration of cell walls and membranes, sa cell wall, we know that it maintains the integrity of the cell. And once this is disrupted, it cannot prevent the cell from bursting due to osmotic effects. So, yung cytoplasmic membrane contains cytoplasm and controls passage of chemicals into and out of the cell. And once this is damaged, cellular contents leak out. And for viruses, damage to the viral envelope interrupts viral replication, no? And note that non-enveloped viruses have greater tolerance to harsh conditions. Okay. For protein, we know that extreme heat or certain chemicals causes denaturation of proteins. And once proteins gets denatured, hindi na siya babalik dun sa original niya na structure. This includes your enzymes. No, pag sinabi natin proteins, this also includes enzymes. Okay, nucleic acids can be damaged or destroyed by chemicals, radiation, and heat. So your DNA, nucleic acids includes your DNA, no? DNA and mRNA. Ay, DNA yun. DNA and RNA. So this damage in the nucleic acids can produce mutants and also cease protein synthesis through the action of RNA. Okay, we now go to the mechanical agents. So we have filtration, yun, physical removal, no, and we can also take advantage of the osmotic pressure. So for us to be able to test, no, the efficacy of a control agent, we have to do the following. So one, you have to expose your organism to the agent. For example, if you want to compare your 70% alcohol versus the 90% alcohol, di ba? Ay, teka. Sorry. Pwede nyo bang i-chat si Angel? Nanghihingi siya ng password. Bakit kaya? Correct password.
Okay
daw.
na, ma'am.
Nag-okay. Ara ka na, Angel?
Yes, ma'am.
I'm sorry. Hindi ko na nakita. Okay. So you have to expose, no, your microorganism sa 70% alcohol and sa 80% alcohol. And then after that, no, you remove the agent or you remove the alcohol. And then put the organism in a favorable growth media. And then check mo if that organism will grow. If hindi siya nag-grow, meaning, or ibig sabihin nun, effective na control agent yung ginamit mo na agent. Okay? How would you measure antimicrobial activity? So we use the MIC or the minimum inhibitory concentration. This is the smallest amount of agent needed to inhibit the growth of a test organism. Medyo mahaba itong MIC, no? But let me read this one. Antimicrobial agent susceptibility assay using dilution methods. So this is a series of increasing concentrations. So bale, for example, class, you have a suspected antimicrobial agent from your green shell or your green mussel, for example, and then you extract you extracted that agent from your green mussel, and then you want to try or to test its antimicrobial activity. So for example, you have a 100 ml of that extract, no? So yung gagawin nyo, di ba sa lab we did serial dilution? So i-dilute mo ngayon yung 100 ml mo. So for you determine the concentration like this one sa ating sample. Ayan. And then you culture a microorganism dyan sa plate mo. And then lagyan mo nung disc or ang improvised na disc for this one is antibiotic or antimicrobial disc is may imagine yung puncher. 'Di ba pag may paper ka, may filter paper ka, ipa-puncheran mo siya, you'll be creating like this one, 'di ba? And then you soak this doon sa iyong pin-repair na dilution nung iyong antimicrobial agent which you extracted from your green mussel, for example. Na-imagine nyo guys yung sinasabi ko? You have your extracted fluid from your green mussel. And feeling mo, meron siyang antimicrobial activity. Okay, so now, meron ka ditong concentrated and then meron ka dito na dinilute mo siya. Ayan, dinilute and diluted further. Now, you wanna test anong concentration yung mag-exhibit siya ng antimicrobial activity. What is the lowest concentration that it will exhibit antimicrobial activity? So maglalagay ka ng mga napuncheran mo na or napansyaran mo na filter paper dito, kasi yung filter paper i-absorb niya yun, di ba? And then meron kang lawn of microorganism dyan, or wala pa siyang growth, no? Ilalagay mo dyan yung mga disc mo na of different concentration, and then papagrow ka ng microorganism dyan, and you check after 24 or 48 hours, anong concentration, yung minimum concentration na nag-start siya mag-exhibit ng antimicrobial activity. Na-imagine nyo, class, yung sinasabi kong MIC? Hello?
Yes, ma'am.
Oo, sumagot kayo. Yan.
Pero medyo lost lang ma'am yung sa punch sa antimicrobial plate.
Kasi meron kasing binibili na antimicrobial disc. Ito, yung ginagamit na puncher. Yung sabi ko, paano ka gagawa ng ganitong mga circle na to? So, yung filter paper, puncheran mo. Yung, di ba, may napupush nyo doon. Pag inopen mo yung puncher mo, di ba may mga bilog-bilog na papel? Ito yun sila.
Okay, ma'am. Thank you.
Oo, tapos ilo-load mo sila dito sa microbial, antimicrobial agent mo na dinilute mo. Okay? So, kukuha ka ng, ang tawag kasi dito is disc. Antimicrobial disc. Tatawagin mo siyang antimicrobial disc kapag nalagay mo na siya doon sa iyong antimicrobial agent. Ito kasi tawagin natin silang antimicrobial agent. So maglalagay ka dyan ng mga disc mo, and then maglalagay ka dito ng halimbawa, one disc from this one, lalagay mo dito. Siyempre may label yung disc mo. Dito, maglalagay ka ulit dito, and then you put here, ayan, ikot-ikot, ayan. And then magpapagrow ka ng microorganism. So uniform, dapat spread plate yung gagamitin mo na method. Okay? Spread plate. Para you will be creating a lawn of microorganisms. After that, titingnan mo saan dyan yung nagkaroon siya ng parang halo or portion na walang grow. Tingnan mo anong lowest concentration na ginamit mo, yung mayroon siyang halo. So halimbawa ito disc mo to, antimicrobial disc, magpo-form siya ng growth sa palibot. Okay? So yan yung malalaman mo na ay meron siyang antimicrobial activity. Ito pala yun. Ito palang slide na to. Okay, so ito yung zone of inhibition. Yung sinasabi kong halo. So tingnan mo, yung hexachlorophene has no effect sa E. coli. Pero ang chlorine, effective siya na antimicrobial sa E. coli. Ayan. And ito, tingnan mo. Pero, si hexachlorophene, effective siya sa Staph aureus. And hindi rin siya effective sa Pseudomonas. Okay? So, what can you say with this one? Dun sa hexachlorophene, it is an effective antimicrobial for gram-positive. Kasi gram-positive si Staph, gram-negative silang dalawa, wala siyang action. Okay? So may mga specificity din yung mga antimicrobial agents natin. So again, ito yung tinatawag na zone of inhibition. The portion around your antimicrobial disc that has no growth. Ayan. So kapag may growth kasi ito eh. Pag may growth, ibig sabihin, hindi siya effective na antimicrobial agent. Okay ba tayo doon? Clear ba tayo doon? So you will be learning more about this diffusion method or this diffusion assay in your laboratory class. Okay. So ito yung relative susceptibility of microorganisms. Ang nasa taas are most susceptible and yung nasa baba are the most resistant. Ayan. So, pag may tanong na gram-negative bacteria are more susceptible than gram-positive bacteria. So, di ba false na yun? Okay? So, ayan. Detecting microbial contamination in food or fish. So, there are different methods that we can use in detecting microbial contamination in fish. So, I guess, pwede nyo na itong basahin na lang yung remaining slides, no? So these are the common foodborne pathogens in which alam ko na hindi nyo sila makakalimutan kasi you made or you are making a video on this microorganisms. Tama ba ako? Hindi nyo makakalimutan yung ginawa nyo na video na microorganism.
Yes, ma'am.
Oo, 'di ba? Kasi ang tagal niyo siyang ginawa, tapos pinag-isipan, tapos nag-ano pa 'yon, nag-involve pa ng inyong editing skills and video video editing skills, ganun. Sige, pag may time tayo, turuan niyo akong gumawa ng ganun, ha. Ano lang yung alam ko basics. Ayan. So, ayan, we can do molecular methods, no? PCR detection. We can also do spectrophotometric methods. So, medyo i-elaborate ko lang itong PCR, no? Yung amplification. Ano yung purpose ng PCR? Alam niyo ba yung PCR? Familiar ba kayo doon? Di ba yung ano natin, one way to determine if a person is infected with COVID-19 is what we call the RT-PCR. Di ba? Narinig niyo yon?
Yes, ma'am.
Meron tayong antibody na test, and we also have the RT-PCR. And mahal yung RT-PCR, right? Kasi mahal yung reagents niya. Alright, ano yung ginagawa ng RT-PCR? Anong ginagawa ng PCR in general? So you are taking a sample, for example, and then you extract the DNA, no, from that sample, and you very minute or very konti, maliit yung makukuha yung DNA from your sample. Kahit maliit or konti yung DNA na nakuha nyo doon during extraction, PCR will amplify or will make thousands and millions of copies of that DNA fragment during the polymerase chain reaction process, and you will be able to detect that during the products during electrophoresis. So yan yung process ng PCR, and meron kang denaturation process, and then a series of ano nga yun, annealing, elongation, and terminate, and annealing temperature, elongation, and then parang termination ng process mo. So paulit-ulit 'yun para siyang imagine niyo na lang na 'di ba ang microorganism or bacteria natin mabilis mag-multiply. Itong PCR process, mabilis din niyang i-multiply yung iyong copies of DNA. Kasi para rin siyang bacteria na yung naunang na-form is exponential din yung growth or yung increase in number of your DNA copies kasi yung naunang na-form na DNA will also serve as the parent para mag-form ulit ng another DNA. Parang ganun siya. So you are using a primer here, and that primer is very specific for the fragment of DNA. So kaya pag parenting analysis, pag gusto mong malaman kung sino yung father or mother ng batang ito, you use a primer for that one. And then, yon merong i-compare yung DNA nung parents para ma-determine kung yun, siya nga ba yung parent or hindi. That's how interesting PCR is. And for the RT-PCR naman, reverse transcriptase na PCR for the detection of COVID-19 virus, we also have a primer specific for the COVID-19. So pag nag-amplify ka, hindi human DNA yung ina-amplify mo na copy ng DNA, but the DNA of the virus. And yun yung madidetect. Tingnan nyo dito, may PCR products. Yun yung madidetect during electrophoresis. All right? You will learn more about PCR when you take genetics. Nag-genetics na ba kayo? Oh, end na pala. Nag-genetics na ba kayo, class?
Wala, ma'am.
Okay, sige. Mag-genetics din kayo soon. Okay. So, tingnan ko yung let's go to module 9. Teka. Module 9. Hindi ko kasi inopen lahat kanina agad kasi mako-confuse ako. Ayan. Saan mo share? Kasi pare-pareho yung template ng slides. Kaya siya, confusing kapag inopen ko yung tatlong lectures ng sabay. Okay, so this is module 9. We have 29 slides here and we have what? 30 minutes. Okay. May class ba kayo after nito? Okay. This slide summarizes yung mga before and after. Noon, ngayon, ayan. Kasi with the use of technology, food biotech actually changed a lot, no? It fueled a lot of changes in the industry, be it in the food industry, in the fishery sector, in the environment. Madaming nagawa yung biotech. Okay? So before and after food biotechnology, maging specific tayo, food biotech tayo ngayon. So before medyo dependent yung crops natin sa pesticides, but nowadays konti na lang yung gumagamit ng pesticides because of the GMO plants, no? We also have better animal health, no? Kasi that's also because of biotech. And we also have better crops. And ayan, better, hindi naman better, kundi less emission of carbon dioxide to the environment. And this one, people now are earning more because of biotech. And we get to have access to a more nutritious food, no, because of biotech. And we will learn why in the next slides. Okay. So these are the top 10 genetically modified foods. So we have the number 1, BT corn, the soy, and all of this down the line. Okay. I-explore nyo na lang bakit sila naging top 10 na genetically modified foods. So for fisheries, no, we have here a genetically engineered Atlantic salmon. Look at this one. Before, ganito lang siya kalaki, but using biotech, no? Through biotech, we were able to grow our salmon up to 5 or 6 times of its original size. Okay. So what is biotechnology? It's the use of living organisms or their parts to make or modify products or improve plants, animals, and microorganisms. So we have the traditional biotech. For food processing, we have the fermented products, mga beer natin, cheese, yon. For agriculture, we have the modifications of the living plants for improved yield. So ayan, meron tayong artificial selection and hybridization of crops. And we also have the direct use or isolation, simple process, direct use of or isolation from original biological sources or fermentation. So ayan, tingnan nyo na lang ito class, no? So with the use of recombinant DNA technology, and what do you know about recombinant DNA technology? Sabi dito, Mr. Stanley Cohen and Mr. Boyer perfect the techniques to cut and paste DNA. So can you imagine? No, they studied DNA and they learned that we can actually cut and paste DNA. Anong ibig sabihin doon? If you have a certain organism that has a dominant trait, for example, sa crops na lang. Yung crop is, it can withstand drought, no? Yung crop na yon. And you want to incorporate that trait doon sa plant mo na mabilis siyang maapektuhan ng drought. So, pwedeng halimbawa sa palay. So, you get that portion of the DNA that's responsible for coding anti-drought or resistant gene resistant to drought, and then you add that to your organism, and then yun yung tinatawag na cut, and then you paste. Ipe-paste mo dun sa organism, and then you allow that palay to reproduce carrying that recombinant DNA, no, sa kanya. And then pag nag-reproduce doon, madami na yung recombinant mo na palay. And you will be producing a lot of palays that can withstand drought. So yun yung recombinant DNA technology. Simple. I hope I have explained it in a simple way kasi medyo complicated din si recombinant DNA technology. Ayan. And you also have, for you to be successful in the use of this technology, you have to have knowledge in cell biology, genetics, molecular biology, and others. Ayan. So, but take note that with the help of this recombinant DNA technology, marami tayong advantages na nakuha sa kanya. Okay, so this is the scope of biotech, no? We have the genetically engineered animals, genetically developed animal vaccines, human vaccines, and then biocontrol agents against pests and diseases, protoplast fusion, embryo transfer, genetically engineered plants, okay, and everything down the line. Okay, so why do we go for biotech? Ang naka-highlight dito is it's the answer for global population and high food demand, no? Kasi we can actually increase our crops and produce better quality and higher yield crops, not just for crops, but also for fishes, especially for aquaculture fishes, because of the advent of biotech. Okay? So ayan, meron tayong microbial biotechnology, manipulation of microorganisms, no, such as yeast and bacteria to create better enzymes for a more efficient decontamination processes, and used to clone or produce large amounts of important proteins for human medicine. Ayan. And we also have useful metabolites from fungi, no? Basahin nyo na lang dito, So, Saccharomyces cerevisiae is one of the most commonly studied strain of yeast, no? And it is microorganism responsible for the fermentation of a lot of products. Okay. So we have microbes here that are being used for a wide variety of products for everyday application. So we have the rennin, okay? Okay, medyo binibilisan ko lang class ha. Okay, so we also have therapeutic proteins, recombinant insulin in bacteria. So I don't know if nalampasan na natin yung insulin, pig insulin or insulin from animal and insulin from humans, no? Mas cheaper na ngayon yung nakukuha nating insulin from human, aside from its being more effective, no? As an insulin para sa mga diabetic people. Mas nagiging mura siya because it's now easier to produce. No, you just scientists took a gene a gene responsible in producing insulin in human, and then nilagay nila yon sa microorganism kasi we know that microorganisms multiply very quickly. Inad nila doon sa genome ng microorganism, and as the microorganisms increase in number, nai-increase din nila yung insulin. Kaya doon ini-extract yung insulin from those microorganisms na pinapag-grow. So microbial growth is not always a disadvantage. At some point, or in most instances, advantageous din siya. If we just know how to manipulate our microorganisms, we can actually produce a lot of useful products from them. Okay? Okay, so we have here a BT corn. Ito yung popular na genetically engineered na crop, no? Do you know what BT stands for? So BT stands for Bacillus thuringiensis. No? BT gene is transferred from the bacillus into the corn. And then, ito yung pest, no? Yung European corn borer. And pag kinain niya itong corn, and ma-ingest niya, no? Ma-ingest niya yung protein encoded by the BT gene, mamamatay itong corn borer. Bakit kaya namamatay yung ano, yung corn borer upon ingestion of the corn that has been genetically modified? And bakit hindi siya harmful sa human? Anybody who wants to answer? So yung BT gene, no, has a protein that kills the corn borer. Specific lang yung protein na yon sa insects. Hindi siya, walang receptor yung ating stomach for the protein para magkaroon ng effect sa humans, no? And as to date, wala pa ring reported na adverse effects sa human health itong BT corn. Okay? Okay, some applications of microbial biotechnology in food processing. So we have here microbial biocolors. Okay? We also have microbial bioflavors. We also have antioxidants of microbial origin. So we have the glucose oxidase, no, the only commercial enzyme used as antioxidant for the removal of oxygen in conjunction with catalase. We also have microbial polysaccharides as stabilizers and thickeners, no, yung alginates, and yun. So, what can you say about this slide? So, ang title nito, Marine Microbes in Various Biotechnological Applications. So, anong pinapakita? So, we can actually use our microorganisms, no, in producing a number of products. Okay? So, we can actually extract bioactive compounds from these microorganisms and be able to produce pharmaceutical products. Pag sinabi mo kasing bioactive, no, when you say bioactive compounds, these are compounds that can be extracted from food material or food from fishes, for example, or microorganisms. And these bioactive compounds provides benefits to humans other than by simply providing nutrition or nutrition. Ibig sabihin nun, meron siyang antimicrobial, yung mga parang meron siyang therapeutic effect or meron siyang function other than by simply providing you nourishment. Ayun. So pwedeng yung bioactive compound mo has antimicrobial, anti-diabetic, yung mga anti natin, no, anti-inflammatory, antioxidant, ayun, matagal kang tumanda if you take a lot of antioxidants, ganun. And we can also produce probiotics or produce talaga antibiotics from these marine microorganisms. And we can also get useful proteins and peptides, no, from them. Pigments, yun yung sinabi kanina. Ito, extract functional compounds like polyunsaturated fatty acids, yung mga DHA natin, yung mga omega-3, you know. And then we can get also useful polysaccharides and monosaccharides. And we can use these microorganisms in the genetic modifications. Ginagamit natin sila to modify certain organism. Okay? Ayan. So we can actually extract antibiotics from marine organism. Interesting ito na research. Okay? We can also extract natural peptides and compounds with antimicrobial activity from marine bacteria. And I have a friend, no, he's studying in Taiwan. He is trying to extract bioactive compounds from fungi. Yan, kasi ano siya, he studies fungi. So biotechnology in fish, no, in aquaculture, aquaculture refers to the raising of finfish or shellfish in controlled conditions. And aquaculture now is taking advantage also of the biotechnology. Ayan, meron tayong mga genetically engineered organisms, yung mga disease resistant na strains of oysters. We also have developed vaccines against viruses, no, that infect salmon and other fin fishes. And yung biotech talaga in aquaculture is widely used in the improvement of the strain of aquaculture species. So sabi nga namin yung joke noon, can we possibly use biotechnology in producing milk fish that has no bones? Yung mga spines, no spines. Pwede kaya yun? Sana pwede, no? Kaya lang, we don't know ano yung magiging effect nun sa milk fish. Baka hindi siya ganun kasarap pag wala siyang madaming spines, no? Ayan. Ayan. So, eto. Note that ito yung first approved, no? FDA approved genetically modified fish for human consumption. So, microorganisms as a tool, we can produce microbial enzymes. Itong TAC, DNA polymerase, is from an microorganism or bacteria called Thermophilus aquaticus. Kaya siya tinawag na TAC. No? It's from a hot spring. Thermophilus, ibig sabihin, it can survive at a very high temperature. Siya yung ginagamit when you do polymerase chain reaction. You add the TAC DNA polymerase, no, during PCR, pero despite being an enzyme, hindi siya na-denature at very high temperature. Yan yung pinakamagandang purpose niya, no? That you can use it during polymerase chain reaction kasi hindi siya na-denature at high temperature. Kasi sabi ko kanina, di ba, during PCR, meron kang denaturation na temperature, meron kang annealing and elongation, and paulit-ulit yun. And until such time that you'll be able to come up or to produce enough number of copies of DNA that will be visible, no, when you run an electrophoresis. Ganon. We also have bacterial transformation. Ayan. So ito yung process in producing human insulin. So yun yung sinabi ko kanina na tinatransfer mo siya dun sa, ito yung recombinant DNA, no? With the insulin gene from human, i-add mo siya sa bacteria. So you use the same molecular scissor para i-cut itong plasmid ng bacteria and yung iyong insulin gene para pag pinaste mo sila, they will fit. Okay? And then you transfect that into a bacterium. Papasok siya dito and every time mag-multiply or mag-grow ito, dala-dala niya itong human insulin na gene. Ayan. Can you see that one? So, di ba mabilis dumami yung microorganism or yung bacteria? So, ibig sabihin, mabilis din dadami itong insulin. That's why nagiging cheaper na ngayon ang insulin with this technology. Na-appreciate niyo, class, yung advantage ng biotechnology? Hello? Nakikita niyo? Na-imagine niyo itong sinasabi ko about insulin? So, you take a portion of an insulin gene from human, i-add mo siya, ika-cut mo itong plasmid ng bacteria. Itong blue kasi bacteria to eh. Ito, ipipaste mo yung insulin gene dito sa plasmid ng bacteria, i-insert mo ito sa bacterial cell, and then the bacterial cell will increase, diba, in number, and dadami sila, at makakaroon ka ng insulin na madami. And i-extract mo ito ngayon, yung insulin mo. There's also another way of extracting the insulin. Extraction and purification of the insulin from the recombinant bacteria. Ganon yun. Yan, meron din tayong molecular cloning. And that's all for Module 9. Hala, natapos natin. And we still have 10 minutes. Are you still there, class? Andyan pa ba kayo, mga bata?
Yes, ma'am.
Hi, salamat. Ayan.
Yes, ma'am.
Okay, let's have a game. Hindi naman siya super game. Sige, pero para na lang, para maging masaya naman yung ating last day of discussion, let's have a game. Hindi na siya quiz. Nag-share ako dito sa chat natin ng link, link to my mural. Please access my mural using this link. Okay? Now, I will share my mural. Ito siya ngayon. So, dapat ma-access. Can you access this now?
Not yet, ma'am.
Pero nakikita niyo yung screen ko. Meron ako nakita si visiting cat, visiting crab, visiting turtle. Sino to? Oh, ayan. Si Jel yung visiting, I don't know, crab. Si Jel yung visiting crab. Bakit yung iba walang pangalan? Ayan. So, for the instruction, nakapasok na ba lahat?
Loading pa po sa akin, ma'am.
Okay, antayin natin. Sige. We can finish this in less than 10 minutes. Okay, for the instruction, you list the common antimicrobial agents that targets the cell wall, DNA, and ribosomes of the bacteria. So, you can actually, hindi kayo makakasulat eh. You go to, you right click, you do right click, no? Mag right click kayo, and then can you see this? Add sticky note. No, you can actually add a sticky note and you can change the color of your sticky note here. So, pwede kayong mag-change ng color, depende sa kung anong gusto ninyo. And then, for example, ayan, increase ko yung size. And then you type. You type your name first. Ako, si Mayflower ako. And then, for the cell wall, ayan, example lang. Anong antimicrobial agent ang pwede kong gamitin that targets the cell wall of the bacteria? Ano kaya? Pwede kayong mag-browse sa internet. Pwede kayong maghanap. Open notes ito. Ito, maglalagay ako ng bacitracin. Ayan. Tapos lagyan ko ng brief description what bacitracin is. Okay? O, i-click mo ito, edit. Ayan. Ayan. So, ako, I answered cell wall, and then bacitracin disrupt gram-positive bacteria by interfering the cell wall and peptidoglycan synthesis. Ayan. So, ano naman ito? Open notes, class. Can you... Ayan. Gusto nyo ba ibaba ko ng konti yung figure? So, you can actually change your color, ha? Pwede kayo dito mag-add. Can you see me? Pwede kayong mag-add dito. You can use the circle, a 5x3 sticky note, or 3x3. Ayan. So, ako siguro, I will change my color. I'll change my color into purple. Okay, pink ako, pink. Ayan. So, pwede kayong pumunta dito sa gilid.
Pwede rin ba na same na sa wall din? Pwede ba?
Ano?
Din na sa wall din, ma'am.
I'm sorry, I can't hear you, but You can answer you can have the same answer. Ayan. But it's important that you put your name and then what part of the bacteria are you going to target using your antimicrobial agent. Ayan. So, ayan. I hope you'll be able to make several sticky notes.
Ma'am, yung mural, ma'am, is naglo-load din lang talaga sa akin.
Ano?
Naglo-load lang, ma'am, yung mural na app. Hindi siya ma-open.
Sa'yo?
Opo.
How about the rest? Okay ba? Okay. Baka sa internet connection, no? If you can't, hindi ma-load yung mural sa inyo, you can just send your answer via our chat. Via chat dito sa ating Zoom.
Okay, ma'am. Thank you.
And then ako na yung gagawa nung inyong you indicate your name first and then kung anong part nung bacteria ang tina-target ng inyong antimicrobial agent. And then ako na gagawa ng inyong sticky note. Ayan. Sabihin niyo na rin kung anong color gusto niyo. Ayan. Wala pala akong video kanina. So I'll give you para hindi naman kayo masyadong ma-pressure. I'll give you 3 minutes. Too much ba? Sige, kasi 2:56 na. I'll give you 3 minutes siguro to finish. Nakabantay ako dito sa chat. Maria Luz, nahihirapan ka sa Nahihirapan ka sa mag-load ng mural? Okay, pasend na lang dito sa chat. Okay, are you done? Oh, kung madami kayong gustong isagot, walang problema. Okay, and later we will share this sa group chat natin, yung picture ng mural, no? Sige, inggitin natin sila. Sana si Shwet. Okay. Violet, Okay. Ay, si Shwet. Daptomycin. Anong tina-target nito, Shwet? Anong part ng cell? Sige, gawin kita ng violet na circle kasi walang naglagay ng circle. Ay. Hindi ako marunong paano siya i-edit. Teka. Ito siya. Okay. Ito yung kay Shwen. Sino pa? Ay, langgit. Sino pa? Si Luz. Nakasend ka na ba? Si Daniel. Daniel K. Anong portion ng cell yung tina-target nito? And ano yung color na gusto mo? Okay, cell wall and cytoplasm. Cell wall and teka, i-paste ko dito para isang Anong color, Daniel K, ang gusto mo na? Ay, anong color ang gusto mo na sticky note? Sige, ako na lang magde-design. Parang walang kumuha ng green. Green ko kayo. Yan. Ay. Ay, Daniel K. Ayaw niyang mag-ano. Ayaw niyang Bakit kaya ayaw? Okay. Anong masasabi niyo sa ginagawa natin? Ang gulo, no? Ay, ito na si Shwet. Ito pa rin siya. Bakit kayong kay Shwet kanina na-copy? Ito yung kay Janine hindi. Kita pa natin. Teka. Sorry class, naglo-load na rin ako. Naglo-load. Loading na rin siya. Synthesis of bacteria. Target cell wall and cytoplasmic membranes. Liitan natin ng konti. Shred, ang dami. Teka.
ma'am, pwede mapadalom gawa ang
Ano?
pwede mapadalom gawa ang page, ma'am.
Ito. Okay, ayan na siya. Okay lang ba yung size? Ayan, sino pa yung hindi ko na? Demi. Ano ba hindi? Demi. Ginawa ko sa'yo. Wala, natula. Ayan, orange. Sige, pagbibigyan kita, orange. Ayan. Ay, damo. Nagaling orange na. Ina lang.
Mama, ako na ma'am, orange, ma'am.
Kay sino to?
Angel, ma'am.
Okay. Change natin. Change na lang kami. Sige, dark na purple. Are, hot pink. Ay, red. Grabe. Sige, okay na. Kay Shred, puro purple. You can also add lines if you want to, or other, ayan, ibang figure. Try ko nga rin. Shapes. Ayan, if you wanna add something like this, ako na. Ay, ang kay Shred, puro lang siya puro lang siya purple na circle. I can see someone moving our main picture. May naghahulag sa inyo. Shit. Shit, ari ka di? Okay. Oops. So, antibiotic. Okay. Pwede kayong mag-araw-araw, ganyan. Aba, si Feline, meron siyang pentagon. Ayan. Pentagon and click for manifesting circle. Ayan. Sino itong manifesting circle? Pwede itong i-delete. Ayan. Angel, wow, damo. Pwede mo siya, Angel, butangan sa... In-move ta ka, Natasha. Pwede mo siya, Angel, butangan sa... Arrow? Something? Okay. Pwede ka mag- Okay. Pwede ka mag-add ng... Ito mo na. Tapos... Ay.
Paano magbutang arrow, ma'am?
Gani, gina-try ko man. Drag. I-drag mo siya, oh. Click and drag on the mural to connect objects. Ayan. Sino to nag-add sa arrow? For example, ano na eh. Tapos, i-increase mo ang size. Ayan. Something like that. Okay. Dapat nag-practice kita, Danay, no? Okay. Doon nag-enjoy na ka mo. Hindi nyo na-feel. Nag-overtime na tayo, class. Ito si Luz. Ayan. Pakapin ito. Oops. Gagawan ko si Luz ng diamond. Dito na. Dito napunta si Diamond. Ayan. Si Shwet man siya po, ang ginakapi yan. Chelsea. Wala siya kapati. Sige. Okay, this will be my last, no? Loose. Ribosome. Sige, later I'll change the color. Na white siya. Blue. Ay, hindi siya gali magpati. Hindi na lang ni gali. Yung mga unique na shapes pala, ano siya? Ay nga, ang ari na green man. Nga ang sa akon, wala.
Okay na mi, ma'am.
Okay na? Sige, tapusan ko lang ng kay Luz. Laba-laba, nagali Luz, oh. Nagreklamo. Hindi siya ma-paste. Hindi ko lang siya basta-basta ma-paste. Ay. Lang get. It's already past 3. Sayang man yung ano ni Luz. Amino glycosides. Tetracyclines. Ang gina-target niya is ribosomes, right? So, amo na lang na danay news. Okay, so isa-isahin natin. Ano lang, very quickly. So, you read your answers, class, no? So, everybody can hear what you've written in our mural. No? Kasi baka pag ako yung nag-read, hindi ko siya masyadong makita. We still have a lot of chats here, no? Medyo madami. Pero hindi kasi siya mabilis i-copy and paste. Anyway, thank you, Daniel, Shwet, and Luz, no? Huwag kayong magtatampo, hindi namin malagay lahat. Okay? Who owns this one? This is quite small. Can we increase the size of this one? Okay, so let's start with, sige, Daniel. Okay. Please read your answer. Oh, wala na siya. Sige, ako na. Daniel says she chose something that attacks the cell wall and the cytoplasmic membranes. Mali yung spelling. C, cytoplasmic. Ay, ano ba yan? Ayan. So it's sulfanilamines inhibits folate synthesis of bacteria at initial stages. Ayan. So what is the importance of folate? So wag na natin i-elaborate. Let's just read your answers. And then I will also read my answer. I think I used pink for my answer. I choose bacitracin. No? This disrupts gram-positive bacteria by interfering with cell wall and peptidoglycan synthesis. Ayan, sino pa? Si Julianne. Julianne says, for cell wall, bleomycin inhibits the synthesis of cell walls in susceptible microbes by inhibiting peptidoglycan synthesis. According another answer from Julianne, targeting the DNA, rifamycins, tama ba yung pagkakabasa ko? Rifamycins interfere with bacterial DNA-dependent RNA polymerase. Then for filine, ribosomes, clindamycin, ayan, disrupts protein synthesis by interfering with, may nag-move na naman ang ating picture, with the transpeptidation reaction which thereby inhibits early chain elongation. Ayan. From Feline, targeting DNA cyprofloxacin kills bacteria by stopping a bacterial enzyme called DNA gyrase. No, from working. From Julie Ann. Ayan. May nagbo-move ng ating picture. Okay, targeting the ribosomes, microlides, antibiotics, inhibit protein synthesis. From Feline again, ribosomes, the doxycycline binds to a ribosomal subunit preventing amino acids from being linked together. Meron nag-answer ng hey, cutie. You like it? Sino yung naglagay nun? Diba? Ang saya. From Nancy, cell wall, targeting the cell wall. She said glycopeptide antibiotics. From Sweat is linezolid or linezolid. Antibiotic used to treat certain serious bacterial infections. And from Luz, nakaka-edit ka na. Ayan, targeting ribosomes. So, ang answer niya is tetracyclines. From Natasha, chloramphenicol, targeting the ribosomes. And ito, yung madaming answer. Si Angel, targeting the cell wall. Gusto niya talaga yung cell wall, dalawa. Autolysins and the carbapenems. Tama ba ito? And also, targeting the ribosomes. Angel answered pectinomycin. And also, the targeting the DNA, quinolones and fluoroquinolones. Quinolones. Ganun. Medyo mahirap din i-pronounce yung mga pangalan ng mga antimicrobial agents, no? Sino yung hindi nabasa? Pakibasa na lang ng answer mo. Si MC, levofloxacin. It blocks bacterial DNA replication. Ayan. So, the next time you will be prescribed with antibiotic or antibiotics, no? You read what that antibiotics is and you read paano niya mako-cure yung iyong infection. So, you read kung ano, does it directly kills the bacteria or the infectious agent, or it disrupts the cell wall, it targets the DNA or the ribosomes. Ayan. So, yan yung parang ano, forever kong iiwan sa inyo, no? Sa ating, because of our Fish 154 class, no? I-share, bale, forever na gagawin nyo yan. Every time na ipe-prescribe kayo with antibiotics, you check, paano magwo-work yung antibiotics sa inyo? Ano yung action niya dun sa infectious agent ninyo? Okay? So, anything that you would like to share? Or anything that you want to say before we end? Before we end our Fish 154 class.
Ay, ma'am, gusto lang naman mag-thank you for teaching us this.
Pasipay biyon ko. Okay, sige.
Guys.
Okay, thank you also.
Yung baliktad.
May pa-effect.
Baliktad ako.
Thank you. Ala, na-appreciate ko gano'n. Ala, this is my first class na nag-amuno. Oh my God. Thank you. Ay, wala lang kamo kabalos ang struggles ang inyong faculty. Ala. I'm so happy.
Thank you so much, ma'am.
Wala kamo
Thank you,
kabalos
good
ang
ma'am.
struggle na.
ma'am.
Thank you so
So
much.
happy. Ala, thank
you,
you very
ma'am.
much. Thank you very much. I hope natuduan taman kamo sa inchakto. Okay. Sige. Wala. How I wish kita subong and tubangay kita subong, no? Thank you very much, and thank you for attending our synchronous classes. And I'm really thankful and happy na kamo akong students. No? Wala kita nagakita na face to face. Wala, ka-cute! Thank Thank you very much. Ano daw, hindi ko gusto i-end ang class.
Picture, ma'am, picture.
Okay, pa-picture kita, everyone.
Naka-share screen gagapon, ma'am.
Oo, wala, hindi niya makitaan. No. 1, 2, 3. Smile. Hala, ka-cute sa inyo. Another one. Smile. Hala, thank you, class. Na-appreciate ko, guys. Ay, lagi ko nag-expect may muna eh. May pa-thank you ka mo. Thank you very much. Nakakataba ng puso. Wala pa naka-submit sang sa inyong video. Ayan, I'm still waiting for your video and ang hambal ko up to the last day of classes. Thank you very much. Thank you very much. See you again sa akong future classes, no? I hope
Thank you, ma'am.
magkita-nay kita sa face-to-face na nga session. Thank you. Be safe and enjoy the rest of the SEM. Ang announcements ng exam, ihatag ko lang sa group chat and sa LMS. Thank you very much.
Thank you, ma'am. Thank you, Gil.
Thank you, ma'am.
Bye.
Super
Thank you, ma'am.
considerate na.
Bye-bye. Thank you. Thank you sa nakaisip sa idea. Oo, wala ka mo kabalo kung paano ako ka-happy. Thank you, Git.